Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Gujarat SET exam question paper for the subject Life Science. This is paper 2 I am discussing. It was conducted in the year 2024. So coming to the first question, in a population the frequency of recessive allele is 0.3. According to Hardy-Weinberg equilibrium, what is the expected frequency of heterozygote? Okay, so they have given Q, that is 0.3. So P you can find, it is 0.7. Frequency of heterozygote is 2pq, so you get 0.42. So the answer is option C. C, D also they have given 0.49. Be careful with the options. So 4-2 is the answer. Then in plant, meiosis take place during sporogenesis and form 4 haploid spores. What is the significance of this division during reproduction? In general, they have asked what is the significance of meiosis. So what is the significance of meiosis? It will maintain the same number of chromosomes in the organism. So what is it? It is responsible for growth and development. No. It helps maintain the same number of chromosomes. Yes, that is correct. Then next one. A heterozygous tall plant, that is capital T small t, is crossed with a homozygous recessive dwarf. This is the recessive dwarf. So small t. What is the expected phenotypic ratio? A simple +2 question. So capital T, small t, these are the gametes. Small t, these are the gametes. You get one tall, one dwarf. So the answer is option A. Then crassulacean acid metabolism in plants minimize water loss in arid environments by. So what is the CAM mechanism? In the night, stomata are open. It is seen usually in the plants which are growing in less water environment, and there is high temperature during the day. So to prevent transpiration, stomata are closed during the day, and at night they are open. So at night, carbon dioxide from the atmosphere will enter the plant. It will be fixed by the PEP carboxylase into malic acid, that is a 4-carbon acid, and this malic acid will be stored in the vacuole till daytime. Then in the daytime the stomata are closed, so no carbon dioxide entry. This C4 acid, that is malate, in the vacuole will be decarboxylated to form pyruvate, which is a C3 carbon atom, and carbon dioxide will be released. So this carbon dioxide, it will enter the Calvin cycle and starch will be produced. Whereas pyruvate, it will help in the regeneration of this PEP carboxylase, which is needed for the next cycle at night. So how does this minimize water loss in arid environment? So opening stomata during day. No, stomata daytime closed. So A is not correct. Fixing carbon dioxide at night and storing it as malic acid for use during the day. Yes, B is correct. A is wrong. Using C3 pathway during the day. No, there is no C3 C4 pathway in CAM mechanism. Preventing photorespiration by directly converting carbon dioxide into glucose in mesophyll cells. That is also not correct. It doesn't prevent photorespiration, it minimizes photorespiration. So that's also not correct. B is more correct. Then the concept of ecological niche can be described as, so it is role and position of a species has in its environment. Role, okay, how the species survive, reproduces, it will adapt to the biotic and abiotic factors in the environment, all these things. Or if you take the example of bee, its role is pollination. The flower it visits and its interaction with the predators, all these things is the role and position of bee in the environment. So answer is option B. Then which of the following characteristics is incorrect? Among the molecules carboxypeptidase, chymotrypsin, elastase and trypsin. They are all proteolytic enzymes. Yes. They are synthesized as inactive precursors. Of course, chymotrypsinogen, trypsinogen. After proteolytic cleavage, they get converted into chymotrypsin, trypsin, all these things. So all are activated by proteolytic cleavage. Yes. All require metal ions. No, only carboxypeptidase A require metal ion. It is zinc ion that is required to function as a cofactor for its catalytic activity. Whereas chymotrypsin, elastase and trypsin, they are serine proteases, and they have got that triad in their catalytic site, that is serine, histidine and aspartate amino acids, which are required for their catalytic activity, but no metal ions are required. So D is the answer. Then which of the following biomes is characterized by low temperature, low precipitation and permafrost? It is tundra. Permafrost is mainly found in tundra. So C is the answer. Then in a recent study, researchers found that certain plant species has a very limited geographic range. Very limited. So this means it is endemic species. A is the answer. Cosmopolitan distribution means found everywhere. It is not cosmopolitan. Is it means extinction risk? No, it is not in extinction risk. Only thing, it is found in a very limited region. That's all. Then in a food web, trophic cascade. Okay. What is this trophic cascade? It refers to the top-down effect that predator have on populations of organisms at lower trophic levels. Yes. So top-down effect means what? You have these, okay, take the case of an ecosystem. You have got the producer at the base level. Above that you have got the herbivores, and above that there are the carnivores. So carnivores are at the top. So they are the predators. So the top, the effect that this predator is having on the population of organism below its level, that is on the herbivore and producer population, what this predator is having, it is called as the trophic cascade. Now suppose an example I am telling you so that you will understand. So we have the predator, you have a grassland ecosystem where the cattle are grazing. So cattle are your herbivore. Then there is a lion also there who is going to eat these cattle. That is the predator. Now what I do is, for example, I will add more lion into that particular ecosystem. What it will do? It will start eating the herbivore. As a result, herbivore number declines, plant population increase. So predator is having a effect on the population of organism at lower trophic levels. Okay. Now the other way around, you remove the predator. Now herbivore will flourish, plant population will decline. Okay, so again the predator is having some effect on the population of herbivore and producer which are at lower trophic level than the predator. That is trophic cascade in a food web. Then next one, ubiquitin. It is a protein, a small protein of 76 amino acid, and it is the process where the ubiquitin, it is attached to the target protein with the help of certain enzymes, and finally it is being taken for proteasomal degradation. So the various enzymes involved are the E1, E2 and E3. E1 is your ubiquitin activating enzyme. It will have the ubiquitin on it. And E2 is the ubiquitin conjugating enzyme. E1 will transfer the ubiquitin to E2. Now E3 is the ubiquitin ligase. It will help in transfer the ubiquitin from E2 to the target protein. That is what E3, it is a ligase that is doing. So when you repeat this E1, E2, E3 cycle, a chain of ubiquitin will be attached to the target protein and finally proteasomal degradation of the target protein. So what is the answer here? It is a quinine. It is a heterocyclic compound. Yes. Polypeptide that gets attached to the target protein by ubiquitin ligase. That is correct. So C is the answer. Then next one, which of the following is a best example of stabilizing selection in evolution? Okay. First one, longer neck in giraffe. Okay, see here. It is directional selection. That means only one extreme. This red one, it is the original population. The blue one is after the selection. Now, so in the original population, there were short neck, intermediate neck, long neck giraffes. Now, what happened? Because of directional selection, that is, it favored only the one extreme trait. Here it was the longer neck of giraffe. So it gave a reproductive advantage for the longer neck giraffe, and the selection is directional selection. Then higher survival rate of intermediate birth weight in humans. Yes, stabilizing population means it is also a natural selection where the intermediate phenotype is favored than the extreme phenotype. So intermediate phenotype will be more in number. So if you take the case of birth weight in humans, so if there are babies which have very high birth weight or babies which are very low birth weight, then those babies are going to have complications. But if the birth weight is around at the average value, then the survival of the baby is more. So high survival rate of intermediate birth weight, it is an example of stabilizing selection. Then next one, increase in the number of black colored moth during industrial revolution. Again directional selection. There were two moths, dark and the light one. Because of industrial pollution, tree bark got black colored. Black moth escaped predation. White moth died in the hands of the predator. So what the environment did? Okay, we will make the dark moth to survive and the white moth will become extinct. So the darker moth survived, and because it was escaping from the hands of the predator. So it is directional selection. One extreme is selected. Speciation in Darwin finches, it is disruptive selection. That means the extremes are selected. See here. The intermediate is not selected. That is either those with the very large beak or very small beaks will survive. Darwin finches. But intermediate beak, no option, less chance. So B is the answer. Then next one, which of the following behavioral adaptation is most likely the result of sexual selection? Camouflage in insects, it is natural selection, because natural selection, it will favor the trait that will help in the survival of the organism. So for camouflage in insects means they can avoid being eaten by their predators. Bright plumage in male birds, yes, attraction for the female birds. Yeah, that cock is having bright plumage, that means he is healthy. Female bird is happy and it will mate with the male bird. So it is sexual selection. B. Cooperative hunting in wolves. If you have watched Discovery Channel, you will know what I am going to tell. If it is a single wolf running behind a prey who is running at a faster rate than the wolf, will the wolf be able to catch the prey? No. But if there is a pack of wolf means many number of wolves, or a group of wolf is there, then this whole group can surround this fast running prey at a faster rate than a single wolf and capture it and have it as their food. So it is also a natural selection. Then migration in birds, again natural selection, that is also for the purpose of survival. That is, birds migrate to escape the harsh environments. That is, if it is very cold, they will migrate to tropical or subtropical regions. So migration of birds is also a natural selection. Then next one, which of the following statement best describes the genetic phenomenon of epistasis? It occurs when one gene influences the expression of multiple phenotypic trait. So one gene, different phenotypes. Only one gene is influencing this phenotypic expression. That is pleiotropy, not epistasis. So next one, it occurs when one gene masks or suppresses the effect of a gene at an other locus. Yes, at an other locus is important. That is epistasis. That involves the expression of both the alleles at a locus in heterozygous. It is codominance, and it refers to the blending of two phenotypes in a heterozygous organism. It is incomplete dominance. Okay. Now next one feature of mitochondrial inheritance is that the disorder. Okay. Mitochondrial inheritance is also called cytoplasmic inheritance. Here the genes are not transferred from nucleus but from other organelles other than the nucleus, such as your mitochondria or chloroplast. And it is uniparental inheritance, that is, it is mainly transmitted from the mother to the daughter or the son. Okay, so it is uniparental. Okay, now see the option is passed from mother to all her offspring regardless of sex. Yes. And mitochondria, it is usually transmitted from the cytoplasm, and the cytoplasm you get from the egg cell. So it is the mother who is always transmitting this mitochondrial inheritance. Okay. Then the next one is passed from father to son but not to daughters. Again, it is Y-linked inheritance, not mitochondrial. Father is only having the Y chromosome which he transmits to sons only but not to daughters. So B is also not correct. Is inherited only if both the parents are affected. That is coming under your autosomal recessive diseases. Follows the same pattern as autosomal dominant trait. I don't know whether any other type of inheritance is there which has the same pattern as autosomal dominant trait. I don't know. I don't think it is there. But anyway, that is not the answer. A is the answer. Then which of the following best describes convergent evolution? Convergent evolution, no common ancestor. So different species, they have the same characteristics because in a response to similar environmental conditions, not because they have arrived from the same common ancestor. No common ancestor is there in convergent evolution. Okay. Now see the options. Mutation that introduce the same polymorphism into two species, okay, after they diverged from a common ancestor. There is no common ancestor in convergent evolution, so A is wrong. The evolution of similar phenotypes in distantly related species. Yes, that is correct. You have wing of bats and wing of butterflies. Both are totally unrelated species, but they have wings for flying. So they have similar phenotypes. So it is convergent evolution. The finding that there are parallel gene duplication in two unrelated species, it is your parallel gene, parallel evolution. It is not convergent evolution. Having only synonymous substitutions in their genes. No, that is not convergent evolution, because what is this synonymous substitution? That means it is a substitution in your DNA sequence in such a way that the protein, the amino acid produced from this DNA sequence is not altered. That means if it was alanine earlier and after substitution alanine itself is produced, it is synonymous substitution. No change in the amino acid. Now since there is no change in the amino acid, will the protein function change? No. The protein is going to function as it is. Then will it result in convergent evolution? No, because convergent evolution wants development of new traits in a distantly related species for adapting to similar environmental conditions. So there should be substitutions which are not synonymous. So at the molecular level, that is at the gene, the changes have to be there in the amino acid production. Only then you can get convergent evolution, not by having synonymous substitution. So D is incorrect. Then next one, which of the following set of statement is correct? Most speciation is thought to occur via allopatric speciation. Correct. Allopatric speciation means through two populations getting separated because of physical barriers, that is mountain barrier or a river, etc. That is allopatric speciation. One species getting separated into two because of geographic barriers. Yes, that is correct. And most speciation is thought to occur because of this allopatric speciation. Then next one, polyploidy is an example of sympatric speciation. Yes. Sympatric speciation means new species arise from this ancestral population by not getting separated via geographic isolation. Okay. Now, and what is polyploidy? Polyploidy means organism have extra set of chromosome in them. Now, why is polyploidy an example of sympatric speciation? I will tell you. Consider a small area having all diploid species. Some mutation occurred, polyploidy have arisen. Now, polyploidy population will not be able to mate with the diploid population. Chromosome number variation. So, what will happen? Reproductive isolation will start to arise. A new species have arisen. Polyploidy, one species, diploid, another species. No reproduction between them. Both are different species. So, in the same area, two new species have arisen without geographic isolation. So, it is sympatric speciation. So, Q is correct. Geographic isolation can eventually lead to reproductive isolation. Yes. A mountain barrier is coming in between a population, 2 species are there. Why? Because they cannot reproduce. Reproductive isolation is happening. So geographic isolation can lead to reproductive isolation. That is also correct. Speciation cannot occur without geographic isolation. Of course, sympatric speciation is there, which is happening without geographic isolation. So that statement is not correct. So P, Q, R are incorrect. That means option C is the answer. Then next one, the commonly used method for the production of monoclonal antibodies in large quantities. A. Western blotting. Western blotting is to detect and analyze specific proteins in your sample. Hybridoma technology, yes, that is for the production of monoclonal antibodies. How? Here what you do is you will fuse an antibody producing B cell with a myeloma cell. Here you get hybrid cells. These hybrid cells are selected on a hatch medium. In the medium only the hybrid cells will survive. Unfused cells will not survive. These hybrid cells are then cultured and they are what you could tell, they produce large quantities of specific monoclonal antibodies. So hybridoma technology is the answer. What about RNA interference? It is for silencing specific genes to know what will be their function. Polymerase chain reaction for amplifying DNA. Then generating a whole plant from a single cell or group of cells in plant tissue culture is known as, it is called totipotency. In your answer key they have given it as organogenesis. So organogenesis means where plant organs like your shoot, root, etc. are regenerated from an explant. That is organogenesis. Generating a whole plant from a single cell or group of cells, it is totipotency. Okay. Now, what is somatic hybridization means? You fuse two protoplasts of distantly related plant species so that you create a hybrid plant which is having both the traits of these plant species. Somatic embryogenesis, embryos produced from somatic cells, not from your germ cells. Okay. Then next one, improvement of human race by controlled selective breeding between individuals with desirable characteristics. What is inbreeding means mating between relatives, closely related individuals. That is inbreeding. So it increases the chance for homozygosity and there will be expression of disease traits also. So it is not an improvement of human race. Then come euthenics. It is improving the human race by bringing improvements in the living conditions as well as in the environment. Genetics means you know, study of gene hereditary variations, etc., etc., etc. Then eugenics is the answer. Answer is option C. Then in environmental biotechnology, which of the following techniques is used to remove heavy metals from contaminated soil? It is phytoremediation. So here they will use the plants to remove or degrade or stabilize heavy metals from contaminated places, because they will be utilizing the plants ability to absorb these contaminants, that is your heavy metals, through the root, then process them in their tissues using various enzymes or convert them into less toxic compounds. So phytoremediation is the answer. And what is microfiltration? It is uses a membrane to remove suspended solids, bacteria and other large particles from liquid, and it is used in reverse osmosis and wastewater treatment. What is bioleaching? It is a mechanism utilizing microorganism to extract metals from their ores. Then biostimulation, it is a bioremediation technique. What is bioremediation? Bio means utilizing living organism. Remediation, to clean up environment with having hazardous contaminants. So biostimulation, it is also a bioremediation technique, and what it will do, it will stimulate the natural microbial community so that this microbial community will produce certain enzymes or some compounds that will degrade the contaminant in that particular area. Then next one, which of the following method is used to identify specific DNA sequences by hybridizing them with a complementary probe is southern blotting. Northern blotting, it is using RNA, not DNA sequences. It is RNA sequences. Western blot, it utilizes proteins, and polymerase chain reaction, it is for amplification of DNA. Then which of the following best describes intermediate disturbance hypothesis? So if you see option B, it tells moderate level of disturbance can foster greater species diversity than low or high level disturbance. That is the IDH hypothesis. Now I will tell you by giving an example. You think of a forest. Now low disturbance, what it means? In the forest there are few dominant species of trees. Now what they will do? These dominant species, they will grow fast and they will outcompete other plants, thereby leading to low diversity. That is low disturbance, not much effect it is causing by disturbing that plant community. Okay. Now what is high level disturbance? Frequent wildfires in that area. What will happen? Okay, wildfire has come, a patch of barren land is created, ecological succession is going to begin. Before it begins, another fire is coming, ecological succession also wiped out. So wildfire, it is preventing the species to establish in the environment. Frequent wildfires, that is high level of disturbance, and it will result in lower species diversity. Now come to the intermediate disturbance or moderate level of disturbance. Moderate level means tree fall due to lightning. So a small patch, you have a barren area. Tree have fallen there because of lightning. Or it may be because of a small fire, again a small patch of barren area. Now what will happen in this barren area? Ecological succession will start. Those which are fast growing plants, they will succeed very fast and they will colonize this small patch of land. And what is happening to the border of the patch where the border of this patch, there will be other plant species which are recovering from this disturbance. So what do you get? You get a mixture of species at different stages of development. So you have the plant colonizing the barren area, plant species which are colonizing the barren area, as well as on the surrounding this patch region you have got the other species which are recovering from the disturbance. So you get a greater diversity there. Okay. So B is the answer. Then next one, cyanide poisoning will immediately deplete cells of NAD+. So what is this cyanide poisoning doing? It is affecting or targeting the complex 4. So what will happen? It will block the transfer of electrons to the terminal electron acceptor, that is your oxygen. And what will happen as a result of that? It will block the entire electron transport chain. Now we know that the electron transport chain, the complex 1 is responsible for converting NADH to NAD+. Now here there is, because of cyanide poisoning, there is blockage in the flow of electrons. NADH will keep on accumulating and the NAD plus will be reduced. Now as a result of this drop in the NAD plus level, the citric acid cycle enzymes will not function. Why? Because you see over here there are 3 steps in the citric acid cycle which require NAD plus. So NAD plus is reduced means citric acid cycle will not function. That means your aerobic respiration is blocked and the cell will have to switch over to anaerobic metabolism. So cyanide poisoning will immediately deplete cells of NAD+. So the answer is option A. Then next one. Protein-protein interaction in live cells can be detected most efficiently by live cells. Remember, live cells, not lysed cells. Okay. Now first one is co-immunoprecipitation. What happens? Here this is the protein in the cell. There are A, B, C proteins. A protein has got a tendency to bind to B. Remember that. Not to C. Now what you do? You lyse this cell. So it is not a live cell. Okay. Co-immunoprecipitation is also for protein-protein interaction studies. Here you prepare the cell lysate, then to that you add antibody. Antibody is resin immobilized antibodies are added. Then you precipitate the antibody protein complex. Now this antibody, it will bind to A protein. Okay, since A protein is bound to B also, when you precipitate out this antibody protein complex, or I can what will happen is this antibody, it will pull down the B protein which is attached to the A protein also. Okay. So this will help to identify which protein interact with each other in a cell. So protein interaction is studied in co-immunoprecipitation, but not in live cells but in lysed cells. Then next option what they have given, it is Western blotting. Here what you take, it is for studying specific proteins in a sample. So here you have the protein, you run it on a page gel, then you transfer the protein onto a membrane, then blocking, washing with the primary antibody, then you incubate with the secondary antibody carrying your enzyme, then you add the substrate for the enzyme and you will detect bands where the primary antibody has bind. Primary antibody will specifically bind to your protein of interest. So that is Western blotting. It doesn't tell regarding anything regarding protein-protein interaction. Then comes the enzyme linked immunosorbent assay. It is for quantification. So here you have the antigen being coated onto the wells of the ELISA plate. Then you add a specific antibody, that is your primary antibody. After that you add the secondary antibody which will bind to the primary antibody, and it will have an enzyme bound to it, and you add the substrate for the enzyme, you get a colored solution, and the intensity of the color, it will be directly proportional to the concentration of antibody present in the specimen. So that is enzyme linked immunosorbent assay. It also doesn't tell anything regarding protein-protein interaction. Now final one is FRET, for Foster resonance energy transfer. It is for protein-protein interaction in live cells. How it works? So FRET, it is tells that this fluorescence energy, it can be transferred between two fluorescent molecules when they are very close to each other. So if you take the CFP, that is cyan fluorescent protein, and YFP, that is yellow fluorescent protein. So CFP and YFP, they have overlapping spectra. That means the emission spectrum of CFP, it will be close to the exciting wavelength of the YFP. So CFP will emit a particular wavelength of light which will excite the YFP, enabling it FRET and making YFP to fluoresce when they come too close to each other. So here CFP is the donor and YFP is the acceptor. So CFP is attached to protein A and YFP to protein B. When protein A and B come together, CFP and YFP also come together. CFP is getting excited and it will release fluorescence which will excite YFP, and YFP will fluoresce. And what you can see here is that there is a decrease in the fluorescence of CFP, and that is the donor molecule, and increase in the fluorescence of YFP, that is the acceptor molecule. So you can conclude that A and B are interacting with each other. If they don't interact, there is no FRET and no fluorescence. So answer here is option C. Then which of the following technique would be most appropriate for determining the three-dimensional structure of a membrane protein at atomic resolution? Okay, atomic force microscopy, it is for visualizing the surface topography of the protein. That is the external domains the protein is having, not the internal ones, external domains of the protein. All that you can detect by atomic force microscopy. And cryo-electron microscopy, of course, that is the answer, because it can resolve the three-dimensional structure of the membrane protein, and it can tell about the position of the hydrogen atoms, and it can reveal the alpha helices and beta sheets also. And cryo-electron microscopy, it doesn't require any crystallization as in X-ray crystallographic technique. So cryo-electron microscopy is the right answer. X-ray crystallography require crystallization and high protein concentration, so it is not used. NMR spectroscopy, you have to go for the solid state NMR methods, not just NMR spectroscopy alone. So B is the answer. Then gene expression across the entire genome can be studied by RNA sequencing, because here all the RNA transcripts can be analyzed. Whereas in a real-time PCR, only one or few genes can be analyzed, not the entire genome. Northern blot also same way. Western blot is for proteins. Again, genes which express only one or two proteins can be analyzed, not across the entire genome. Then next one, in which of the following animal groups do you find organisms that are acoelomates? So acoelomate means they lack the true body cavity, that is coelom. So it is found in Platyhelminthes. They have a solid body filled with mesenchyme. They are acoelomates. All the other are coelomates. So the answer is option B.